Hello everyone, and welcome to the sixth episode of the Health Talk series organized by Pune Knowledge Cluster. I am Dr. Preeti Nema, your host for today's talk. Pune Knowledge Cluster is one of the six S&T knowledge clusters established by the Office of Principal Scientific Advisor to the Government of India. Our mandate is to bring together research organizations, industries, civic authorities, and the citizens to foster the science, technology, and innovation ecosystem in the region. To know more about our programs and activities, please visit our website. The link has been pasted in the chat box. And at PKC, we run several community engagement programs, including citizen science, citizen-centric science talks on various themes. One of which is the one that you're attending today, which is the PKC Health Talk series, which primarily focuses on spreading awareness about public health and also aims to educate the citizens about the recent technological advances in biomedical sciences. Today's episode brings to you an introduction to clinical genomics, which has revolutionized modern medicine. Clinical genomics aids in prediction, diagnosis, and treatment of complex diseases like cancer, metabolic disorders, and rare genetic diseases. And to talk to you about this, we have with us today our esteemed guest, Dr. Ramesh Hariharan. Dr. Ramesh Hariharan is an adjunct professor at the Indian Institute of Sciences, Bangalore, and is the CEO and founder of the Strand Life Sciences company. This is a research and diagnostic company that applies next generation genomics to generate valuable insights into genetic diseases, facilitating drug discovery and personalized healthcare. Dr. Ramesh is a recipient of the TR100 Award of Young Innovators by MIT's Technology Review magazine. He also received the Global Indus Technovator Award from MIT, which was instituted to recognize the top 20 Indian technology innovators worldwide in 2003. Dr. Ramesh has got his academic and research training in computer sciences from IIT Delhi, the Max Planck Institute of Informatics, Saarbrücken, and the New York University. At Strand Life Sciences, Dr. Ramesh leads a multifunctional team that works on the interplay between computer science, biology, and medicine to further genomics research and to build genomics applications for disease diagnosis and treatment. Welcome, Dr. Hariharan, and thank you so much for taking time to talk to our audience. Over to you.
Thank you so much, Dr. Preeti, and I'm glad to see a sizable audience here, and I hope that I can tell you something interesting over the next 45 minutes. Let me share my slides. Are my slides visible?
Yes.
Yes.
Okay, perfect. So clinical genomics is the act of applying genomic information, which comes from DNA sequencing, um, to decision making for healthcare. So you could be diagnosing disease, you could be helping treat disease, etc. For the most part, I'm going to focus on diagnosing disease. And if you look at how we diagnose disease today, you have a problem. You don't know what it is. You go to a doctor, the doctor will do some basic physical measurements like blood pressure, pulse, etc. So we are used to these simple devices for physical measurement. And they have their utility and they go a certain distance. If that doesn't help solve the problem, the doctor would then take some blood and send it to a lab where you would do some assays. Typically you look for certain molecules in the blood. So these are called clinical chemistry assays or immunoassays. And if you find a high LDL level, your doctor might prescribe statins or something like that. So this is one way of one more tool for diagnosing disease. And then if that also doesn't help, then and things look more serious, then um, the other tool that we have is we've got imaging. We do scans. Typically, these images involve some form of ionizing radiation, not always, but many times, and they give you some sense for what's going on internally. Um, Between all of these sorts of approaches is where we were until about a decade ago. And then over the last decade, uh, a new tool has been added to the box. And this new tool allows us to get deeper into the human body at the molecular level and understand what the molecular entities there, in particular nucleic acids, which include DNA and RNA, and their various sister species. Uh, what is really happening with them? So we can actually today sequence nucleic acids in the body, and that gives us a new tool which allows us to access more of the state of the body than could be done by imaging or by simple clinical chemistry assays or by physical devices. But it also has a lot of complexity, and I want to walk you through examples of how this is useful and the complexity involved. And before I do that, of course, notice that nucleic acids, there are many different types of nucleic acids. There's DNA, which which is familiar to us as a double-stranded structure of the type that you see here. DNA gets converted to RNA. RNA is a single-stranded structure, which is also nucleic acid. So you can sequence the RNA as well. You could sequence the DNA. And then RNA gets converted to proteins. Proteins are three-dimensional objects that engage in various chemical reactions, and the dynamics of those chemical reactions often determine the state of what a cell is doing or not doing. And you can also do sequencing, strangely enough, to measure the state of the proteins, even though proteins are not nucleic acids. So today you can do sequencing at multiple levels and get a understanding of what is happening at every level. And that gives you a window into looking at disease in a much deeper way than we could have done a decade ago. And that has allowed us to um, solve a number of what are called diagnostic odysseys. There are people who have had unexplained diseases for several years. So here, this is one case from our practice. There was neuromuscular degeneration for many years, and that means progressive loss of control over skeletal muscles started at the age of 27. And very often these things happen without any family history. What do you do in such situations? You consult doctors, you go to many doctors, they all sort of throw up their hands because they just didn't have the right tools to um, to look at this problem. And over a period of 16 years, many doctors in India and US consulted, many different hypotheses put forth that match the symptoms of this individual, and no convincing answer until finally, you do sequence the DNA, and sure enough, the answer is right there. There are mutations in that DNA in a particular gene that's instrumental in the synthesis of a particular um, compound called sialic acid, and the deficiency of the sialic acid is leading to progressive disease. So that at least tells you where to look and what potentially to do in order to prevent the disease from progressing further, or even to think about attempting a cure. Um, so these sorts of odysseys are not uncommon, even though they qualify as what are called rare diseases, which means one in several tens of people or one in 100 people thereabouts might have things like this. But um, until 10 years ago or so, it was very difficult to get an answer to these. Today, we've got a much better shot at solving these odysseys. And how do we solve these odysseys? So I want to walk you through the process and get you a good understanding of what it involves. So for the most part, I'm going to focus on DNA or the genome sequencing. As I said, there's also RNA, there's also protein. There are many forms of RNA. You can sequence all of those. They all give you information that the other one perhaps doesn't always give. And so uh, so the genome sequencing, DNA sequencing is not one thing. It's a whole collection of tools. But today I'm going to focus on DNA sequencing because it's the most basic of those tools, and perhaps quite powerful in its own right. So when we talk about the DNA, we are talking about the genome that we are born with, which is about 3 odd billion characters. Every cell in our body, almost every cell in our body has 3 billion characters. In fact, there are two copies of each of these 3 billion characters. So there are actually 6 billion characters in almost every cell of the body. These characters are organized into, you can think of it as a bookshelf where there are many books. Each book is just a lot of ACGTs thrown around. You'll have two books, a pair of books called labeled 1, which refers to chromosome 1, a pair of books labeled chromosome 2, a pair labeled chromosome 3, and so on, all the way till chromosome 22, where there's a pair of books. The, the pair of books here for chromosome 22 is, as you can see, a much shorter, much smaller book than the chromosome 1 book, but nevertheless, it is similar in the sense that it's also a sequence of ACGTs, but a different sequence of ACGTs. And then there is the last chromosome, which is the sex determining chromosomes, where males have two different books really, and females would have the same book repeated twice, the X and the X. Um, and you can see that the Y is much smaller, and there's some reports that evolutionarily the Y is getting even smaller as time progresses. We'll see where that ends up eventually. But the thing to note is that if you take any pair, any chromosome pair, let's say chromosome 10, then the two books in that pair are very similar to each other. They differ only in 1 in 1,000 characters. So this book and this book are almost identical to each other. They differ only in 1 in 1,000 characters, while the chromosome 9 books will be very different from the chromosome 10 books. But between the two chromosome 10 books, you'll have almost identicality, and likewise between the chromosome 9 books. So that's what the genome is about. And our task in sequencing the genome is to get to this sequence, get to this precise ACGT sequence. Because you and I don't have the same sequence. We are different in 1 in 1,000 characters. At a scale of billions, it means that we are different in millions of characters. Any two people will be different in millions of characters, and we need to figure out what those differences mean and whether they can explain any particular disease that one person might have that the other person does not. And how do we read the DNA? So, how do we read the genome? So, we take uh, many copies of the DNA. So clearly one cell has already sort of two copies. And then if you take a whole collection of cells from your blood or from your saliva or anywhere, you'll get a large number of copies. And each of those is a whole bookshelf with many books like this. And the way DNA sequencing works today is that you've got to take these copies, chop them up into small pieces. Now, this is a perhaps an irritant, but it's a necessary irritant. As we'll see, it's not possible to do otherwise. It would be ideal to just somehow get the sequence of this entire DNA fragment from one end to the other without having to chop it up. But today you have to chop it up into small fragments of about 150 each. And then you can go and read each fragment. And what you get through that process is the sequence of each fragment. The sequence of each fragment will be one of these ACGT sequences. So every fragment may have a slightly different sequence or a very different sequence also, because they have all come by fragmenting this larger DNA. And so you get what are called reads or shreds of paper in this picture. Each read will have a different sequence potentially. And now you've got these reads and that you have to put together. How I want to spend one slide on giving you some insights into how does one get the sequence of each of these little fragments. But note the fact that you don't have the sequence of the whole thing as a unified whole. You only have the sequences of the individual fragments, which are all obtained by randomly cutting these larger DNA molecules. So how does one read these fragments and get these so-called reads out of these fragments? Here is a very quick picture. You start with, let's say, these fragments in green. You've got let's take one fragment in green that we want to sequence. What is done is that special sequences are added to both ends. Why those are done, we don't need to worry about that. So there's a green sequence and a pink sequence added. And then you take that entire fragment and attach it to a surface. And then you do PCR, which now, thanks to COVID, is a household word. So you create many copies of these fragments, of the same fragment. This is a single fragment. There were many fragments we began with, but I'm going to talk about a single fragment here. You take a single fragment, attach it to a surface, and then make many copies of it. And now you create what is called the complementary strand. A complementary strand is simply, you know that DNA is double stranded. So if there's an A on one strand, there'll be a T on the other. If there's a G on one strand, there'll be C on the other. So each strand complements the other strand in terms of characters, and the information content in the two strands is identical, just that an A is replaced by a T and a G is replaced by a C. So, in nature, there's a way to take one strand and create a complementary strand, sort of. So it starts at one end and it keeps adding character after character. So every time it sees an A, it'll add a T here. Every time it sees a G, it'll add a C here, and you will keep sort of extending this and creating the. That's what this arrow shows. Starting from one end, there are enzymes that will just create the complementary strand, and you engineer those enzymes so that every time they add a A here, because there was a T out here, you add a A here, it lets out, let's say, blue light. And every time you add G out here, oops, it lets out red light. And similarly for C and T. And you're monitoring this process all the time with an image, with a camera. So as you monitor, you'll get these flashes of light out. And you're not just doing that for one fragment at any given point of time, you're doing that for billions of fragments. So this surface will have geographical regions separated out for billions of fragments. And then each of those fragments, you would have PCR'd into many copies. And then the second strand would be synthesized. And as the second strand synthesizes, light of different colors would keep coming out depending on what characters being added at every point of time. And as you monitor this image, some image analysis can tell you which character was added at each point of time. And that is really what a read is. It tells you the sequence within this entire fragment. And you have that being done in parallel for billions of fragments. And eventually you get a file onto your computer, which is for every shred of paper like this, about length 150, you get the sequence. And you get that in parallel for billions of fragments. So now the challenge is that you had your whole DNA to begin with, which was broken into chromosomes, all right, but only about 23 pairs of chromosomes, or 46 chromosomes, let's say. But now that's been further broken into many much smaller fragments, 150 length fragments. So you've got billions of these little pieces. You've taken a book and torn it up into small pieces of paper. And now you've got to assemble this jigsaw puzzle because our interest is in the original DNA, the entire DNA, and not just in these tiny fragments that have been sequenced. So you get about a billion such reads if you sequence the whole genome, and a billion such scraps of paper. And now we've got to put them together. And how do we put them together? So there are two main principles, one which is harder out here. And the way to do that, the harder way, is that you take two of these scraps of paper. Now, these scraps of paper, I have transformed them into orange bars. But think about each of these orange bars. Like this bar is one scrap of paper from the previous slide. This bar is another scrap of paper from the previous slide. And you're starting to assemble the jigsaw puzzle simply by saying, this scrap of paper has a TCTG at the end, and this has a TCTG at the beginning. And therefore, the two must have come from, they must have been adjacent in the original book. They just got torn apart. So I can put them together. I can just connect them together and create a slightly bigger piece of the jigsaw. So I start piecing together the jigsaw this way. And hopefully, if I'm lucky, I'll just keep piecing them together and I'll get the whole original genome back. The challenge is that that doesn't happen more often than not. Uh, this becomes, because there are a billion such pieces, there are lots of choices. There are a lot of reads which will have TCTG at the end, and many which will have TCTG at the beginning. You won't know which pair came together. So you'll have to navigate your way through the search space. For those of you who are from a computational background, the search space ends up being very large, and it doesn't lead to eventually a unique solution. So eventually what you do is get to as far as you can get, and then you do further experiments to disambiguate all the ambiguities, and keep doing that till you can connect all the fragments together in the right order and put the whole jigsaw together. So that has been done once, and or a few different times it's been done, and that sequence doesn't belong to you or me. It belongs to some random, some arbitrarily chosen 2, 3, 4, 5 people. And we call that the human reference sequence. So that we already have. Some 4 or 5 people were taken. This painful process over many years was undertaken, and then we got the human reference sequence. It is not your genome, it's not my genome, because your genome differs from the human reference sequence in 1 in 1,000 places. Mine also differs in roughly 1 in 1,000 places. And for the purposes we are looking for clinical genomics, we want to take a single patient and look at what their DNA sequence is, which means that we have to precisely understand where that patient's DNA sequence differs from the reference sequence. So, for example, if the reference sequence has an A, but the patient has a T, then this is the place of interest, because the reference sequence presumably is a healthy person's genome. And if we have a person with a problem, then we need to look at how they differ from the reference. So if I have an orange bar that comes from a scrap of paper that comes from sequencing a particular individual, a patient, let's say, how can I save myself from going through that onerous jigsaw puzzle solving approach of connecting scraps of paper together? And the way to do that is to use the reference sequence. You take each scrap of paper, each orange bar that comes from your patient, and you just search for the sequence in the reference sequence. You're not guaranteed an exact match because any two individuals are different, but you know that the differences are small, about 1 in 1,000. So chances are at least most scraps of paper may differ from the reference sequence, maybe in one character, maybe in two characters, not a whole lot. So if your scrap of paper is 150 long and you search for it in the reference sequence, allowing for 1 or 2 mismatches, then you will find a hit. So you can take the scrap of paper and search for it, and you will be able to place it against the reference sequence at a unique place. And if you were to do that for every single scrap of paper, so you take each of these orange bars, which is an individual scrap of paper, and you search for it in the reference sequence, and you place it where it finds a match, allowing for 1 or 2 mismatches, then a nice picture emerges. So wherever you see that, so firstly, each of the scraps of paper, each of the orange bars will get placed at one place in the reference. A small fraction of the orange bars will not even get placed at uniquely at one place. They might find multiple matches. Some will not find any matches. Those are complexities that we won't look at today. Those are real things to worry about. But an overwhelming fraction of the the orange bars or the reads will find a unique home in the reference sequence. And when you place them there, the picture that emerges is as follows: that you see that all of the orange bars here are saying that there's a T, while the reference sequence says there's an A, which means that the patient that we are sequencing has a T at this location where the reference sequence has an A. So these are called variants. And it is these variants in the patient that we want to identify. And as I said, the number of variants in a patient will run into millions. So every single person has millions of variants relative to the reference sequence. So we can identify all of these variants. And that's the first part of the story. And before I move to the second part, uh, I want to bring one complexity out here. It is not necessary that all of the reads, all of these scraps of paper, all of these orange bars say that there's a T. Some of them could say there's a T, and some of them could say there's something else, there's an A. And this happens because we have two copies of every DNA molecule, every chromosome, we have two copies, right? One which comes from our mother, one which comes from our father. And so the two need not agree. Um, Not unusual for spouses not to agree, but in this particular case, as you can see, the one of the maternal copy is saying perhaps there's a T, the paternal copy is saying there's an A, and that's perfectly normal. These two need not agree. And there are variants of this sort. So there are variants that are what are called homozygous. They are common to both copies of our DNA. And there are variants that are present in one copy of the DNA. The other copy is identical to the reference sequence. So we call out both these sorts of, we identify for every patient both these sorts of variants, and there will be, as I said, millions of those. Roughly, uh, a, a, maybe a third to a quarter will be homozygous, meaning they'll occur in both copies of the DNA, and the remainder will occur in one copy of the DNA. So once we have these millions of variants called, then what do we do? How do we diagnose the the cause of disease in a particular person that's come to us for help? And there begins our task of understanding variant impact. Sure, the reference sequence, the healthy genome has an A and I have a T. Now, what does a T really do? It turns out, of course, most variants, since there are millions of them, most variants don't really have a drastic impact. They might make your, um, I don't know, nose a little longer or your complexion a little fairer or darker, or they'll have subtle effects, in other words. While some variants are overwhelming in their effect and they can just bring the whole system down. And we need to find that needle in a haystack, right? Which of the millions of variants is problematic? And to do that, we go through a process. We try to understand what is the genome doing and how do variants impact that. And we just need to understand the genome a little bit to do that. So firstly, if this is the genome, then of course, this is a large, you can think of it as a large featureless landscape of ACGTs, but you can also think of it as more structured. There are interesting features and landmarks all across the genome stream. Some parts of the DNA, some parts of the genome are called genes. So I've just labeled arbitrarily a colored portion here. So they're called genes because they carry the recipe for the creation of a of proteins that we talked about. So the genes will have a code that gets converted to RNA, that gets converted to protein. Proteins interact in chemical reactions, and those reactions are essential for various bodily functions. So the entire large stretch of DNA that we have, 3.2 billion characters, uh, is not entirely featureless. Some small fraction of it, a couple of percent of it, is what we call genes. And these genes, about 20,000 of them, carry recipes. And the rest of the DNA, rest of the genome, has its own controlling functions on the genes. So the rest of the genome does not carry recipes, but it carries controlling functions that determine when is this recipe executed, with what speed is it executed, etc. So for, again, those who come from a programming background, think of these genes as sort of variable descriptions and think of the rest of the genome as control flow that is operating on the variables. Right? The uh, so the genome has this structure. More often than not, we focus on the genes because that's what we understand the best, and see whether there's a change in the recipe, because the change in the recipe is something that we are better at predicting and which is also causes more drastic effects, while changes in the rest of the DNA causes more subtle effects on when is it that this recipe is being executed, etc. Much harder for us to predict today, but not to underestimate the role of the rest of the genome. There are lots of portions that are truly very important, but our ability to use those is still limited, while our ability to use the genes has progressed by leaps and bounds over the last several years. And a lot of the examples I'll talk to you will be about the genes. Now, genes are not laid out in a contiguous fashion in the DNA. They're laid out in an interrupted fashion. So you'll have a stretch of ACGTs that makes the gene, and then there's a long stretch of ACGTs that's not part of the gene. Then again, a stretch of ACGTs that is part of the gene, then another long stretch that's not part of the gene, and so on. So the recipe is contained in these exonic regions, these so-called exons. They contain the recipe for the proteins. The intronic regions, these are called intronic regions, they're also stretches of ACGTs, but they don't carry the recipe. But they do carry signals that delineate the end of an exon and the beginning of an exon, and we'll look at that in a bit. So exons carry the gene recipes, introns do not. And going by the same vein, our ability to understand the exons today is much better than our ability to understand the introns. And so a lot of what we'll do today is talk about the exons, but we will also talk about the introns a little bit. The exons carry a code which we've all known, a famous Nobel Prize with a person of Indian origin is associated with this, Hargobind Khurana. So in the exons are recipes where triplets of DNA characters, so ACGT, so triplets of characters are read and converted to a new molecule called an amino acid. So if you see a TTA, it'll get converted to L, which is leucine, I think. If you see TCT, it will get converted to S, which is serine, and so on. So the code in the recipe code inside an exon is really laid out in the form of triplets. Each triplet is read and converted to an amino acid. And these amino acids are strung together into proteins. Note one of these amino acids is special in the sense that actually more than one, but one is particularly special, that if you have a TAA or a TAG, it gets converted to an X. Now, X is not an amino acid. It's the signal for the end of the program. So again, for those who are from programming background, uh, there are many programming languages that have a beginning of the program, end of the program. So this is sort of the end of the program. It signals the end of whatever the gene is trying to do. Likewise, there's a beginning, but the beginning gets overloaded with one of the amino acids. That's the end is particularly striking, as we'll see. Um, so now, with that background, I want to walk you through a bunch of cases on how um, these are from our practice and how we solve these cases and what is involved at the DNA level that leads to disease. So here is the first example where there was progressive loss of, so this is somebody who came to us with the progressive loss of central vision. So until their early 30s, they were fine. And then suddenly the eye started deteriorating. The central vision became like this, what you see. This is how you would see somebody's face. Um, this is eventually it got diagnosed as what's called Stargardt disease, but initially it wasn't clear why this is happening. And then when we sequenced, this is what we found. So at the top, you see uh, this is the reference sequence in a particular gene. This is what was there in the patient. Ignore this bottom two bars right now, just focus on the top. So in the reference sequence, there's a C. In the patient, the C had become a T. There was a variant at a certain location. The C had become a T. Now, what happens as a result? So you can see these triplets that I alluded to on the previous slide. So 3 characters at a time are read and converted to an amino acid. The next 3 are read and converted to an amino acid, and so on. So now what happened to the fact that the C had become a T? Then the triplet CGA became TGA. So the triplet changed. And what was the impact of that? Well, TGA stands for the stop marker, which means this protein that was merrily running along has now come to an abrupt end out here. This is a stop has been introduced for no reason whatsoever, except by the complete happenstance of a C becoming a T. As a result, this protein has gotten truncated prematurely and it becomes non-functional. Um, the well, this has happened in one copy. Remember, we have 2 copies of the DNA. So in one copy of the DNA, the C has become a T. So shouldn't the other copy step in and rescue? Now we looked at the other copy and that had in the same gene, there was a G that had become an A. And as a result, the triplet changed. GGA became GAA. And therefore, the amino acid changed. So this amino acid, E, was different from this amino acid G. And as a result of which, the protein actually changed in, I'm sorry, there's some noise out here, but it should die down in a second. The G actually, sorry about that. So the G actually became the amino acid became from a G, it became an E, and that caused a change in the second copy of the second copy of the DNA, and therefore the protein that came from this. And together, these 2 variants completely compromise this particular gene. Now, how do we know that this is a problematic change? There's no easy way to. This one is very easy to predict. You've prematurely truncated the gene. This one is a little harder to predict because there's a change here. And the only way to predict that this is a bad change is go do an experiment. And so people have done these experiments to figure out what happens when G changes to an A in this particular gene, and they figured out that there's a problem. and these are test tube experiments. And what happens as a result is that for this particular gene, both copies are compromised, and this gene is really plays a garbage collection role in the eye. So when the way you see is that there are certain chemical reactions that happen when light hits the eye. Those chemical reactions lead to toxic waste being generated. This gene helps clear the toxic waste. If this gene is compromised, the waste is not cleared. Over a period of 30 years, the waste accumulates. As waste accumulates, the eye cells start dying, and progressively there is greater and greater death, and therefore loss of vision. So, and that's why it sets in after 30 years, because garbage, for 30 years, the eye can take that garbage accumulating, but thereafter the cells start dying. Um, Now, let's move to the next case, where this was a family which had two sisters in their 20s. This time it's not the eye, but the heart. The heart gave up, what is called heart failure, where the heart's ability to pump blood is dramatically lowered. And while in the previous case, we saw that there was a single character changing to another, here we saw something slightly different. Here we saw that in half of the reads, which means in one copy of the DNA, two characters were missing. So A and G, which are there in the reference sequence, they were missing in the patient at hand. While in the other copy of the DNA, things were perfectly fine. Those two characters were there. So this two character deletion in the uh, in these two sisters created a challenge that uh, somehow the other copy of the DNA, even though it was completely, its integrity was completely preserved, could not overcome. So there are these two types of diseases, some in which one copy of the DNA goes back, the other bad, the other copy still manages to recover. While in others, even one copy going bad is bad enough. So in this particular case, this was bad enough for the heart to give up. These two characters deleted was bad enough for the heart to give up. And the reason why the deletion of these two characters was a problem was because you see what happens when these two characters that are there in the normal person, the reference sequence, and they go away in the patient, then what happens to the triplets? You can see that the triplets change completely. So if three characters had gone away, it would have been a little better. Then the triplets, one of these frames would have gone, but the rest would be preserved. But if only two characters go away, then the whole thing gets offset thereafter. And that leads to what's called a frame shift. And that's a very dramatic situation that pretty much makes the gene very different from what it is supposed to be. So frame shifts on account of deletions of or insertions of characters in non-multiples of three are a very catastrophic event in our DNA. And in this particular case, where it really affected is that the DNA has a lot of our heart has a lot of mechanical structures that keep there's a lot of action happening, right? The heart is beating without a stop, and there are these rivets, and there are these rivets that connect things to various fibers, and this part of the connector goes bad, and therefore over 20-25 years of beating, they just come apart and the heart is unable to pump anymore. So faulty riveting, as we call it. The Moving to the next case, we saw a case where there was a single character being replaced by another. Then we saw a case where there's two characters being deleted. And here's a case where there is a large chunk of characters being deleted. So nature is playing games with the DNA all the time, right? So these are all real cases. So a 5,000 long stretch of characters was deleted. And this poses challenges for us when we try to use DNA sequencing to get hold of this large deletion, because the scraps of paper we get from the patient look like this. You see the TGA out here, and and then you see the CAT out here, but you don't see any of these intermediate parts that are there in the normal person, that are there in the reference sequence. The reads or the scraps of paper that come from the patient will just look like this, TGA CAT. And when we try to search for TGA CAT in the reference sequence, we will not find it, because you'll have TGA, you'll find TGA, but after that, there's a whole lot of stuff. And you'll find CAT, and before that, there's a whole lot of other stuff. But TGA CAT, you'll never find. And so this took a long time for us when we were setting up all of the computational processes, that not you may not find these things in their entirety. You need to start looking for prefixes and suffixes. And when we did that, indeed, we found a whole bunch of prefixes lining up in one place and a whole bunch of suffixes lining up in another place, indicating that there was a huge stretch in between that was deleted. So this huge stretch that was deleted uh, eventually led to the diagnosis of in this kid that was born with the most curious thing, where their organs were out of place. The liver is typically on one end and the stomach on the other end, and it was flipped around. And it took us a long time to get all these computational pieces in place to be able to find these really large deletions that also show up. Um, the Moving to the next is something even more complicated. I told you in the a few slides ago that we will focus largely on the exons, but now I'm going to talk about the introns and how they affect things. So this is a case of beta thalassemia. Beta thalassemia is a disease where you need to have blood transfusions every month. It's very painful because your blood doesn't have the ability to carry oxygen. The hemoglobin is compromised, and this is the one of the genes that contributes to the hemoglobin. It's called the HBB gene, and it has only 3 exons. And somewhere deep inside this intron, a T in the reference sequence or in the healthy individual had become a G, somewhere deep inside this intron. And this is the only thing that we could see wrong in this person who came in with a diagnosis of beta thalassemia. The exons were completely intact. And for a while, we were puzzled as to why would something in the introns, which is not part of the recipe, how would that matter? And then, as we read up and figured out how, it led to this fundamental question of how does the cell know that an exon ends here and the next exon begins here? Well, it knows that because there's a GT where the exon ends and there's an AG where the exon begins. And before the AG, there's a stretch of characters that are largely C's and T's, and before that, there's an A in there. So you have all of these motifs that characterize the beginning and the end of the exon. So just remember this, there's an AG at the beginning of the exon and uh, a stretch of C's and T's just before. And what had happened as a result of the T here becoming a G. So this is the reference sequence and this is the patient at hand. And remember the T here had become a G deep inside the intron. And what had happened as a result is that the AG, which is at the beginning of the exon, a new AG got created deep inside the intron. So suddenly the cell started thinking that the exon begins here instead of beginning here. And that added 11 characters to the gene. And when you add characters in non-multiples of 3, we've seen frame shifts in the past. And these frame shifts are catastrophic events, as I said. And that pretty much crippled this gene, leading to a case of beta thalassemia. Um, Moving to the next case, this is my own story, color blindness. So I realized I was colorblind. Most of you can probably spot the number hidden inside these dots. I cannot. And I was curious as to what is really the cause of all of this. And it's a long story on how we got to the answer, but I'll just tell you the answer eventually. And it'll be yet another sort of game that nature has played in the DNA. So most people have DNA that looks like this. There's a red gene. By a red gene, I mean it's a gene that creates a protein that allows us to see red color. And there's a green gene that creates a protein that allows us to see green color. Um, and for some strange reason, there are 2 copies of the green gene in most normal people. So most normal people look like this. And in my DNA, uh, nature had decided to cut and paste and chop and paste, perhaps in when I was created, or perhaps in some ancestor of mine. I'm not entirely sure. But somewhere a cutting and pasting event had happened, which did a really bad job, and it created a configuration like this, where this particular gene ended up being half green and half red. So I have a red gene, I don't have a green gene. I have a half red, half green gene. And these two are not used, unfortunately. The third one here or the third and fourth one here are not used. So I ended up having, most people have a red gene and a green gene. I ended up having a red and a half red, half green. And that led to my color blindness where I can't differentiate between red and green as easily. Right? Um, there are many, many, many other stories, and I'll just allude to one just to give you a sense for why these things are often high stress situations. We had two kids who passed away mysteriously. We did sequencing. We we found there are millions of characters that are different, right? Millions of variants. So through a variety of tricks, we narrowed down to one which we felt was highly suspicious, but we couldn't nail it down. We couldn't say this is the cause, and that's you're going to see a lot of that because so much is unknown today. And the mother was pregnant with the third kid, and the question was should the pregnancy continue. So you can see how difficult this question is. We have a candidate, it's a plausible candidate, we don't have convincing proof, and can we abort somebody on account of something which we don't have convincing proof of, right? So these sorts of situations one goes through quite a bit dealing with the unknowns. The I want to move to end on two cases which are more popular and that lift the application of DNA sequencing from those with rarer forms of disease to slightly more common forms. So Angelina Jolie, you might have all, I'm sure you all know her, and you might have read her blog about 10 years ago exactly. She wrote this blog. Her mother, many members in her family had died of gynecological cancers. So she got tested and found that she carried the BRCA gene. 10% of all breast ovarian cancers are caused by um, mutations in this gene. And then she chose to undertake various risk reducing prophylactic measures. There are many ways to do that. You can monitor yourself more aggressively, or you can choose to have surgery to remove organs in which cancers are likely to form. And that reduced her risk down quite substantially. And since then, she's been, of course, blogging to say, how do women get more access to gene testing? Because this is really life saving. Right? And another example, more recent, well, same time, perhaps actually a little older, but Sergey Brin, who's the founder of Google, um, found that he has a variant in his gene that puts him at high risk for Parkinson's. His mother also has Parkinson's. And so his risk is up from 1% to 30 to 75%. And he's, of course, investing a lot. And so that's where this adage of forewarned is forearmed comes in. You know, once you know, even though there might not be a solution today, you can start following research carefully. You can start doing, of course, if you have wealthy, you can start investing. But as he says, he can get his risk down by about half, um, to about 25%. And um, if there's steady progress in neuroscience, then another by bringing it down by another 13%. So get it down to 13%. So there's clearly a lot that's going to happen as we go along. And I want to spend the last two slides, so a couple of minutes, saying the core problem today that we have. I hope I gave you a glimpse of how it's being used today. But the core problem we have today is that so much that's unknown. You have millions of variants in individuals. We don't know what each variant does. We need better prediction methods. We need better lab methods. We need better ways to scan the literature. These are all key tools that the community is building. And one method by which we can understand all of this better is we sequence lots of people, people at large. Costs are reducing. Millions of individuals have been sequenced so far. The number is growing rapidly. And as we put together large databases of people who don't necessarily have a reason to get sequenced, but as their clinical data and as their genomic data comes together, we will start understanding in much more detail what our genomes really do. And we will see an explosion of this activity over the next decade. And to sign off, all of this is just the DNA, just the genome. As I said, there's RNA, there's protein, and then this is just the genome that we are born with. Our genome changes over our lifetime. And that is a whole world of new applications for a different talk. We will not touch that. That's an ocean in itself. But you can see that there's so much to do and so much more to learn. So I hope that gave you a good glimpse on what genomics is about, what clinical genomics is about. And I'll pause for questions now.
Thank you. Thank you so much, Dr. Hariharan, for such an informative talk, and the the analogy of chromosomes as books and variants as spelling mistakes in the text was so wonderful to understand the whole concept. So we have got a mix of questions, technical and non-technical. Before we take up those questions, I would like to inform the participants there will be a feedback form pasted in the chat box, and you can fill in the form. There are students who have been asking us, do we get certificates for attending this talk? You can get them. Please fill in the this form that will be pasted in the chat box very soon. Okay, so we'll take up the questions. First one, as you were saying that this testing is increasing, one of the questions that has come from the audience in our office is how accessible genome testing is in India. Is it available in small cities?
Yes. So I would say about 5-6 years ago, it was completely in its infancy. Now you will see doctors in definitely tier one, tier two cities becoming increasingly conversant. And uh, as long as you can access a doctor in one of these cities, samples can be picked up from your home, sent to a central lab. Most of the prominent labs are in Bangalore, but it doesn't matter. You send your sample in, report comes back. So it is eminently accessible. You just need access to a specialist, um, and which you can, and now with the teleconsulting and all of that, getting access to specialists all over the country is much easier. Did I answer the question? No.
Preeti, are you in?
Yeah, we can't hear you.
I'm so sorry. I'm so sorry.
Sorry, somehow
Yeah.
the audio broke. After, did
Yeah.
I answer the
No,
question?
no. So I'll repeat the question, Dr. Hariharan.
Yeah.
One of the questions from the audience is how early can we diagnose genetic disorders? For example, there was a person in your patients that who got to know after 30 years, like this is the problem because of a genetic reason. So how early can one get to know that this person has got a genetic disorder?
So technically, the uh, um,
So
the
maybe I'll
DNA.
add another question. Sorry to interrupt, Dr. Hariharan. I'll add another question from the audience. When does a doctor decide that a patient should get genomic testing done? And in that process only, we'll like to ask, like, if a patient has got predisposition, how early can he get it diagnosed?
Yeah. So technically, we're talking about the DNA that you're born with. So you could technically sequence yourself at birth. If the way things are headed, costs of sequencing are reducing, at some point of time, we will see that it would make sense for everybody to just do the sequencing and keep it with them so that over their lifetimes they can refer to it as needed. Um, Now, the second question is perhaps more pertinent today is when do doctors recommend sequencing? I think doctors recommend sequencing. Sequencing has traditionally been viewed as expensive, and so doctors recommend it when on a need basis, when they clearly see that there is something that they can do with the. So, if there is, for instance, a clear onset of disease and you want to diagnose that, that's one instance in which they would recommend. Second is when perhaps you have a family member who has the disease and you want to know whether you are at risk. So often sequencing has been referred to only as a reactionary measure to disease already being there, either in yourself or in your family. And where the world is moving today is to say that you can do a lot of this proactively. There is a lot unknown, so you have to be prepared for that. And but costs are coming down. It's going to just get more and more accessible, and so this is the Earth is shifting at the moment. So there won't be a single answer as we progress.
Yes. Okay. So the next question from the audience, Bhavna asks, which cells in the human body are taken for genetic testing or genome sequencing? Any cell or cell from specific organs?
So what you are born with, you can take any cell. If that the DNA, if you're doing sequencing of the inherited genome, you can take any cell. It could be blood, it could be saliva, it could be a heel prick, it could be a dried blood spot, it could be even hair. I mean, for those of you who haven't realized, the Indus Valley genome, one skeleton from the Indus Valley genome was sequenced recently, a
Yeah.
couple of years ago. So any cell will do. Now, for certain other applications, and I refer to the changing DNA changes over your lifetime. Now, those applications are much more sensitive to where you take DNA from, because the DNA in different parts of your body will change differently over your lifetime. And if that is the cause of disease, then you have to take DNA from those parts. So in cancer, for instance, it's important to take it from the specific organ where cancer is involved, etc. So that's the complex answer to that question.
Okay. Another question from the audience. Anto asks us that can epigenetic changes in humans be found by genome sequencing?
Yes, indeed. So the I didn't refer to it, but one of the things that changes over your lifetime is that while the genes themselves change in more subtle ways, the certain changes of molecules being attached to the DNA, they change in very structured ways over your lifetime, and then they can change in unstructured ways also, and they do change in cancer. So by monitoring how those changes happen, these are called epigenetic changes, you can monitor your process of aging. You can monitor, you can detect cancer early, for instance. So several of the programs that we're working on involve measuring from the blood these epigenetic changes and using those to detect disease early. So those are also sequencing applications, because um, you can convert uh, epigenetics into a genetics based test, and there are clever ways to do that.
Okay, so a question on similar lines by Dr. Madhavi Rane is, can addiction make changes in human genome sequence? She asks.
So, I mean, the addiction, you're saying, addiction as an addiction to alcohol or
Maybe
something.
alcohol or drugs,
Yeah.
maybe.
So, I mean, the Lamarckian view of the world that your environment can change your genes is is generally not considered true, but there is evidence, of course, that your food habits does change your epigenetic profile, and there is a small amount of evidence of how those epigenetic profiles can be inherited downwards. And so there is a small chance that epigenetically you can hand down um, the impact of your alcoholism to your children, grandchildren, perhaps. But by and large, the DNA, the genome itself does not change as a result of environmental influences, but it changes in its own way over your lifetime.
Mhm. Next question is, can genome testing be used to detect or predict neurodiversity like autism and other disorders?
Um, so there are the full spectrum of diseases. If I were to lay those out, many diseases. So there are diseases that have a very focal genomic cause. There is one place in the DNA where one or two characters have gone bad, and that is strong enough to cause disease. And then there's the other extreme where the the disease is strewn across many characters in the DNA and very highly multifactorial, and also multifactorial in the sense that it's while the DNA renders some greater susceptibility, various environmental factors chip in together, and then that causes disease. So all the diseases we know of fall in the spectrum. Diseases which are uniquely and very focally genomic, and diseases which are highly multifactorial, both in the genome and in the combination of genome and environment. Our ability to look at, obviously, the focal ones is far better today. As we go along, we may get better at the latter category. Autism itself, now coming to a specific question, has both ends of the spectrum. There is the focal end of the spectrum where we can actually diagnose cases. That's a fraction of all the cases. And then that's the multifactorial end of the spectrum where we are not so good at today, but we are getting better. And all these population sequencing projects that I was talking about, where we have large scale sequencing, will help us, get better and better at these multifactorial effects of the DNA.
Okay. So the time, we have already crossed the time limit, but I'll take few more questions. One from Jitendra Kumar is, since most of the mutations identified in complex disorders have little effects, therefore variance explained is pretty low. How can we overcome this bottleneck? This is what he has asked.
So he's referring again to the fact that where the genomic contribution is highly multifactorial, so many parts of the DNA together contribute to the disease, and each individual part has a very small contribution, and that's where large data sets come in. So if you have highly multifactorial influences, then you need very large data sets to be able to um, create predictors and reliable predictors from that. And I think we're on a path to that. And as billions of people get sequenced and we have their clinical outcomes as well, we'll be able to create these predictors that handle more multifactorial effects.
Mhm. One last question which I'll take up is by Debapriya Gautam. She asks, the sequencing details from a sequencer does not come in a user-friendly way. It needs a huge downstream processing. So which application do you get to use? Do you use to get a user-friendly report?
There is no there's no single answer to that. Uh, One has to companies such as ours, of course, spend a lot of invest a lot in software. The world itself is investing. There is a lot available in the open source if you were to download and use. It's But you will have to string a bunch of things together and then put in some effort on top of that. So I don't have a simple answer to your question.
Okay, so one more, there's so many questions that are coming in. One question from Dr. Sham Sundaram is, who publishes the human reference DNA sequence? He would like to know more about the human reference DNA sequence that you were
I think
mentioning.
as of now, there are a couple of organizations in the world, NCBI, which is the National Center for Biomedical Information. It's probably part of the NIH in the US, or maybe it's a different organization, I'm not sure, but it's a very prominent organization. Then there's EBI, European Bioinformatics Institute, and these are some of the places where DNA sequences are archived, and including the human reference sequence, as well as for many other species.
Okay, so there are questions just coming out. Last question is, sticky blood disorder, is it heritable?
So by sticky blood disorder, I assume you mean sickle cell anemia,
I think so, yes.
which is where
I'm
the hemoglobin molecules stick together and form these chains, um, and causing the blood cells to twist into a sickle shape, and those cells don't easily go through the blood vessels. And yes, that is very eminently heritable. And well, it says antiphospholipid disorder. I'm not quite sure about that, but um, there are many that are clearly heritable. And anything, as a thumb rule, anything that happens early in life is more likely to be heritable than otherwise.
Okay. So we'll wind up the session now. I see some more questions, but you can email us those questions, and we can send the collated version of questions to Dr. Hariharan and get you the answers. I would now request everyone to switch on their cameras. We would like to take a group photo. As the audience will be switching on the camera, there are some announcements to make. One is that a feedback form has been put in the chat box. Can you put it once again? Gayatri or Sheetal, please help me with that. So the feedback form is there.
Yeah.
And
But Preeti, we have shared a message in a chat box.
okay, so Dipali Nerka says not able to fill the feedback form.
Okay.
So in case somebody can help in.
Sure.
And yes, camera is switched on for many of us. Can we take the group photo? Are we done?
Yes, Preeti. Thank you so much.
Thank you so much. Th- Thank you so much, Dr. Hariharan, for enlightening us about genomics, the clinical genomics and the applications, and taking up questions from all types, technical and non-technical. I see there is a lot of curiosity. We'll be collating questions from the audience, and we'll send you in case there are many. And thank you so much, everyone, for joining in. Please follow us on social media. We keep making announcements of all the talks that we have for the citizens. And it was a wonderful session, Dr. Hariharan. This is Dr. Preeti Nema now signing off. And thank you again.
Thank you very much.
Thank you, everyone. Thank you so much.